Baver, Scott Baver, who's the Director of Global Affairs, will discuss MERIDIAN, a study to evaluate the efficacy and safety of Pegcetacoplan in ALS.
All right, can you see my screen?
Yes, thank you.
So good afternoon, everyone. Thank you very much for giving me the opportunity to come in and talk to you about the work that Apellis is conducting in ALS. So I'm Scott Baver. I'm on the medical affairs team here at Apellis Pharmaceuticals. And today, over the next 10 or so minutes, I hope to give an overview of complement and then segue into the potential role of complement and focus specifically on component of complement C3 in ALS. And then finally, I'll walk through the study design of our ongoing phase 2 study of Pegcetacoplan in sporadic ALS. But before I get started, I wanted to give some important information here and just share with you that Pegcetacoplan, it is an investigational compound. It has not been tested for safety or efficacy with respect of a demonstration of safety or efficacy in ALS, and it's not approved for use in ALS by any regulatory authority. So I'm going to start with a brief introduction to the complement immune system. And I think, as many of you are already aware, the complement system is a part of our immune system that's designed to attack foreign invaders. So if we think about bacteria or abnormal cells in the body, it really does so by setting off a cascade that I'm going to show in the next slide. But suffice it to say that initiation of this cascade, it ultimately results in cell death and then inflammation. So when a person's complement system is what we call uncontrolled, that system ultimately attacks cells in the body that are really critical for overall appropriate physiological function, rather than just fending off foreign invaders or abnormal cells. So one last thing to point out here is that while the complement immune system has been implicated as a major player within innate immunity, the complement system is now really recognized as that key conduit to both innate and adaptive immunity. So what you can see here is that while it seems that the complement cascade is very simplistic in concept, as the cascade of events lead ultimately to inflammation and cell death, the complement system overall is very complex in that there is really just a lot of different players here. So there's up to at least 40 proteins that are involved in the complement cascade. And what you can see here on the top of the slide is that there's multiple different ways to start the complement cascade. And we call these initiation pathways. And what you can see is that there are 3 here: the classical, lectin, and alternative pathways. And really, these initiation pathways, they're activated by specific molecules. But what I really want us to focus on that I think is worth noting is that right here, all 3 of these initiation pathways, they converge on this protein called C3, which we think is central to the process of inflammation and ultimately cell death by lysis or phagocytosis via this opsonization process. So I also want to point out, and think it's important, that C3 activation that we see either here or ultimately going down here, that really results in, again, it results in inflammation, but it also can result in cell death, both at the levels of C3 and C5, which some of you may have heard of being the terminal complement complex. So leaving you at the end is that while under normal conditions, the complement cascade acting normally, it really allows our body to maintain overall health. But in diseases where complement is overactive, this can result in complement system attacking these otherwise healthy cells. And therefore, we think that modulating this complement cascade is a desired therapeutic. So now let's think about this as, what does this mean for ALS? And so while I've walked through this high-level presentation of complement, we're next going to walk through some evidence of how complement may be involved in the pathogenesis of ALS. I do want to stop and say that the most important thing about complement and our understanding of complement in ALS with respect to the pathophysiology of disease is that it may or may potentially only be a part of the underlying pathogenesis of ALS. So numerous pieces of evidence that we've seen that have been published or presented, they demonstrate that inflammation plays a role throughout the course of disease. And these data include a potential role of complement in ALS. So specifically relating to complement, there is evidence that complement components, including C3, it's elevated in serum, brain, and spinal cord of people living with ALS. And while preclinical evidence in animal models of ALS has demonstrated a link between function and C3 concentrations, there's been a recent publication by Jalgaard and colleagues that really showed that function, which is measured with ALFSR, correlates nicely with C3 levels in people living with ALS. And I think maybe more interestingly, the scientists here is that there is literature that suggests that in animal models of ALS, that an overactivation of C3 and specifically C3 deposition at the neuromuscular junction, that this leads to motor axon retraction even before symptom onset. So taken together, at Apellis, we feel that reducing the activity of complement in people living with diseases where there is uncontrolled complement, and we really believe based on these data that we see on this slide, that this is the case with people that are living with ALS. We believe that controlling this complement may lead to reduced inflammatory components of the neuromuscular junction, and then ultimately, hopefully, to cell survival. So this has really led us to the investigation of our C3 inhibitor, Pegcetacoplan in ALS. So Pegcetacoplan is classified as a complement component C3 inhibitor, and it's a twice-weekly subcutaneous infusion that's now approved in the United States for use in adults with a rare hematological condition. It's called PNH, but it's also being investigated in clinical trials for multiple other diseases, including ALS, and in the study that we're about to walk through today. So it binds to the central complement molecule C3 that we just discussed, as well as a breakdown product of C3, which is a molecule called C3b. And what we can see in this schematic here is that by inhibiting C3 and C3b directly, we believe that pegcetacoplan can directly modulate overactivation of complement activity and then really reduce all inflammation initiated at the level of C3 and C5, and then ultimately potentially cell lysis and phagocytosis. Okay, so now that we have that presented, what we're going to do is walk through our design of the MERIDIAN phase 2 trial of pegcetacoplan in sporadic ALS. So the MERIDIAN trial, it's an ongoing randomized double-blind phase 2 trial looking to enroll about 228 individuals living with sporadic ALS. So this is a 2-year study. And so following screening, enrolled participants are or will be randomized 2-to-1 to either twice-weekly subcutaneous pegcetacoplan or placebo for the first year, or what we can see here is 52 weeks. And following the first year, all participants are then able to roll over to a 52-week open label treatment period that will only contain treatment with pegcetacoplan. So the primary endpoint of the study is combined assessment of function and survival, or CAFS, at 52 weeks. And so while our primary endpoint is CAFS, we'll be looking at function with the ALSFRS-R and survival as secondary endpoints. But importantly, we really wanted to determine the role of pegcetacoplan on disease progression overall. So we have CAFS as that primary endpoint here. But we'll have additional endpoints, secondary endpoints that are going to include safety, slow vital capacity, and quality of life that will be measured for both participants and their caregivers. And all measures will be measured over the course of the entire study. So the last slide here is just highlighting some of our key inclusion and exclusion criteria. So participants who are eligible are any adult individual with a diagnosis of sporadic ALS, with onset of ALS symptoms within 72 weeks of the screening period. So in addition, slow vital capacity needs to be of at least 60% of predicted value, and the ALSFRS-R has to be at least 30, both of these at the time of screening. We do allow individuals to be receiving either riluzole or edaravone during the study. They just can't initiate or change their dose for either 30 days, for riluzole or 60 days for edaravone prior to study participation. So again, I want to thank everybody for giving me the opportunity to come and present on the potential role for complement being a targeted therapy for ALS and going through the MERIDIAN study. If anyone wants more information, you can go to the trial website, which is the meridiantrial.com that you can see here. And you can also go to clinicaltrials.gov for more information specifically about the study. Thank you.
Thank you very much. It's very, very intriguing. Let me ask you, so has MERIDIAN started already?
Yes, MERIDIAN has started and is currently enrolled.
Yeah, excellent, excellent. Terrific. I guess we don't see any questions in chat at this point. I guess the one question I would ask you is how you calibrate the probability of success in this approach with some of the recent, and how you reconcile that with some of the recent discouraging results from complement C5 inhibition. Can you just comment briefly on that?
Yeah, so I really try to show how C3 acts centrally to reduce all inflammation as well as cell death, both by C3 specific and C5 specific pathways. And we really still feel confident, even though the disappointing results with the C5 inhibition, that there's still potential for that extra level of modulation at the level of C3 to really provide benefit to the people living with ALS.
Hopefully that's the case. And can you comment just a bit further on the relative preponderance of activation and importance of C3 in the periphery, which you documented so well, and the CNS?
Yeah, it's a great question. And we have active preclinical studies ongoing for that hopefully will have more data to show at an upcoming conference.
Thank you. Excellent. All right. Well, thank you for that.